Recording in progress.
Okay, I should do it. All right, so today's lesson, factors affecting enzymes. So in the previous lesson, and enzymes are made up of mostly proteins, and that they are very sensitive to a lot of changes, right? Their function is dependent on a lot of factors, and they also have a lot of features about them that you guys need to be aware of. So today's lesson is going to talk about how their function can be influenced by a few factors, right? So this is talking about temperature, pH, concentration, and also about the presence of inhibitors and also coenzymes. Okay, so there's four factors that can influence essentially the function of a protein. That goes as changes in pH, right? Changes. There are five factors that have an influence on the function of a enzyme. First one is changes in temperature. Second one is pH. Third one is concentration. Fourth one is whether there are inhibitors present. And then the fifth one is whether there are coenzymes present. Okay, so we're going to look at each of them and we're going to look at what they do. Okay, so first thing, let's do a little review. Can someone define for me in the most simplest term what is an enzyme? All right, can I get Abi, any ideas? Yes, so an enzyme is something that catalyzes a biochemical reaction, right? So essentially, you could just simplify it as saying a biological catalyst. Okay, catalysts are anything that reduce the activation energy for a chemical reaction to occur, and enzymes are basically the biological version of those. Okay, next, where does the substrate bind with the enzyme? Can I get Kevin? Okay. At what place does the substrate bind to the enzyme? There's a specific name to this place.
Binding site.
That is located on the substrate. What's the one on the enzyme?
Is the shape right? Ethnographic.
Any ideas? Someone else? Janita? What do you think? Yeah, it is called the active site. So essentially, you would only, I would suggest, and bindings because it's going to be on the enzyme. And those enzymes, they can change their shape that the active site doesn't allow for binding sometimes. So it's more important that you know the active site and how it can change when it comes to the enzyme action. Well, you're not supposed to see that. Okay, I'll just skip through it. A drug molecule has been designed to inhibit the activity of an enzyme. The shape of an enzyme is shown as below, right? So you can see an enzyme's normal activity and you can see its active site, right? So when that active site is affected such that it doesn't appear anymore, the enzyme loses its function. Okay, an enzyme's function is heavily tied to its active site. If an active site has been blocked or it's changed shape or it isn't the same shape it was, it's losing its function altogether. It can't work properly if the active site itself is not properly shaped. So think of it as a jigsaw puzzle, right? If you have two jigsaw puzzles, even if you cut off one side, it's basically pointless. Both of them will never fit together again because their shapes don't conform with each other. That's the same thing with enzymes and substrate. If the enzyme's active site has been changed, has been affected, then essentially it loses its function. And that's what today's lesson is going to be about, okay? All the different ways an enzyme's active site can be changed or can be basically affected such that it won't have anything binding to it anymore. Okay, so these are all your glossary tasks as always. And yeah, so essentially there are six factors, but these two fall under inhibition. Essentially, it has to do with the binding onto the active site, but they work differently. So these six factors will influence how a enzyme can work. Let's look at the first one when it comes to. Okay, let's do a small catch up of the previous lesson because I realized that we haven't done the lesson. It's been a while since we've done the lesson, so I'll try to go very quickly about enzymes. Okay, so enzymes, they are biological catalysts. They have active sites where a substrate molecule will bind. Okay, these substrate molecules and enzymes are very specific for each other. Okay, one enzyme will only allow for the binding of one substrate. So an example, lactase will only allow for the binding of lactose. Sucrase will only allow the binding of sucrose. Okay, so they are designed, they are built, their molecular structure is such that they only allow that one substrate to bind to them and then be broken down into their products. Okay, like I said, high specificity. There are some enzymes that can have a low specificity where they allow for more common molecules to bind to them, but we are focused on the high specificity part because that's what an enzyme is known for. Okay, so essentially a substrate molecule, that's the big molecules. For example, that's lactose. It will bind to the active site, and imagine this is the enzyme this is lactose. It will bind to lactase, and once it's bound to the active site, it will form a substrate, enzyme substrate complex, undergo a chemical reaction, and then the substrate is broken down into its products, glucose and galactose. Okay, so essentially with the enzyme, it facilitates the breakdown of the substrate molecule. Remember that the enzyme itself does not carry out the reaction. All that the enzyme does is lower the activation energy required for the chemical reaction. So enzymes are not carrying out the chemical reaction, they are lowering the activation energy. That's why they are a catalyst and not anything else. Okay, so another thing you guys should know, you guys would have referred to this word a lot during your EcoLink excursion and also when we talked about enzymes for the first place. But what does it mean to be denatured? Okay, you actually hear this word a lot today as well. When a protein is denatured, basically what has happened is the bonds that are holding that protein together have been broken down and such it loses its original shape. And like I said, once the protein loses its original shape, it becomes useless. A protein is only functional when it has folded into a quaternary or a tertiary or a quaternary structure. So as soon as those structures are broken down and it's becoming a different shape, it loses its function. That is what it means to be denatured, where the bonds between the amino acids have been broken down, which causes it to undergo conformational change that prohibits its intended function. So essentially, the active site has been deformed. Okay, so this is what it looks like, and if it was exposed to a unfavorable environment, then it will become denatured. Active site is no longer going to be the same shape. Then the original substrate cannot bind to the active site anymore. Yeah. So this all is possible because of the specific shape that enzymes have based on their target substrate, right? So you can see on the pictures on the side here, each enzyme has a specific shape for its specific substrate, okay? So depending on the substrate, that active site will be shaped accordingly. And when they bind together, they form the enzyme substrate complex. And as soon as that active site is affected due to heat or pH or any other factor, then the function of the enzyme is also affected. Any questions about this? This is a very important thing to realize, is the fact that the active site and whether it allows for substrates to bind to that active site is how an enzyme can work. If at any point that active site is affected or blocked, enzymes won't work, essentially. Okay, that's what today's lesson is also going to be focused on. So the fact that enzymes have that specific shape allowing for the substrate to bind is referred to as the induced fit model. So what happens is the enzyme binds to that shape, and because it is so specific to that shape, the enzyme substrate will actually change conformationally to hold the substrate tighter. Basically, imagine someone coming in for a hug, and then as soon as they come in, you hug them tighter. That's essentially what's happening here. because it's such a good fit, it causes conformational change at the active site, allowing for a stronger bond to form, and then it will be converted into products and then released again. And remember that enzymes can be used multiple times. They're just going to keep repeating this process over and over until all the substrate has been broken down into products. Okay, so that was what we looked at in the previous lesson. Anyone not sure about this so far? Okay, all right. So today's lesson, we're going to look at how different factors can then influence how an enzyme works. Okay, so the first thing we're going to look at is temperature. So essentially, enzymes are so specific to where they are working, right? For example, inside our body, enzymes are working in a very specific temperature, which is 37 to 38 degrees Celsius, right? That is the range of temperature it works with, and that is the temperature the enzymes work best at. There is a range at which all enzymes work best at. So that will usually depend on their location or function. So one of the things you guys realized at EcoLink was that you were using a enzyme that can function at a very high temperature, right? I think I've included it here. Yeah, here. So one of that enzyme is called Taq polymerase, Taq DNA polymerase, and it can function at a high temperature, which is 75 to 80 degrees Celsius. That is because the bacteria that originally made that enzyme survives in a very hot environment, in hot springs, right? So it needs to be able to make enzymes that survives in that temperature. So it can. But the fact is, enzymes will have a very specific temperature range where they work best at. And you can think in our body, that range is that very much tied to our okay. So enzymes are very temperature sensitive and can easily lose their function if they go within, out of that range. This is because the amount of kinetic energy that the molecules have will affect the bonds that they are forming. So essentially, remember how I said that? So let's have a look here. So let's have a look here. There's two situations, right? One where there's optimum temperature, the active site is fine, the substrate binds, and then the product is released. Another one is where the temperature is too high. This is when there's too much kinetic energy being supplied. The bonds between the amino acids are being broken down. The active site starts to become denatured, and the substrate is no longer able to bind to that site, right? So essentially, the effect of high temperature on enzyme is that it will cause denaturing. So the shape of the enzyme will be forever changed. And this is a permanent effect. Once an enzyme has denatured, it cannot undenature. Okay, the bonds cannot be formed back in the same conformation because it took a very specific kind of folding to make it fold in that right configuration. And once that folding is lost, it can't be regained again, right? So in our body, the optimal range is between 36 to 38 degrees, most effective at 37. That is why when you have a high fever, they actually make sure to cool you down because they don't want your body to experience shutdown. Essentially, if your body increases temperature to a certain degree, 40 degrees, your organs start shutting down because essentially all the proteins in your body start denaturing. They all hate the heat. And as soon as it goes beyond the range, the proteins start breaking down and I can't say this, but all hell breaks in your body. Basically, all your organs are shutting down, right? Because the proteins that make them up are becoming denatured. So functions can't be carried out and things go wrong a lot. So that is what happens when you go above the optimal range. The protein denatured, it won't allow for it won't allow for a substrate to bind to the active site. What happens if you go below the temperature? Essentially, what happens is that the enzyme freezes, right? But it won't lose its function. It is able to gain its function again if you carefully reheat it to its optimal range, but essentially it loses its function because it's been frozen. It can't carry out its function because the enzyme has been frozen. So it is still able to survive if you heat it up, but then again, yeah, it becomes less active until eventually freezing, and it can regain its function again. Any questions about this? So if it goes above the range, denatured, completely lost. If it goes below the range, it's frozen, but it can regain its function. Okay, that's what you can see in this table here, right? In this graph here. So if all proteins, you can construct this graph of showing the optimal temperature of an enzyme, right? So essentially, the lower the temperature, the more it will freeze, right? So you can see the function is very low and slowly increasing until it reaches a peak. This is the optimal temperature at which enzyme activity is the highest, and you can see that's the temperature it works best at. And as soon as it goes above that range, you can see that enzyme activity plummets because enzymes are starting to denature. They're starting to lose their conformational, they're starting to undergo conformational change. Active sites are becoming unbind, the substrate can't bind to the active site. So essentially, it will lose all function. So you should be able to draw a graph like this if once you know the range of a enzyme. What is the optimal range? And then you should be able to draw a graph like that if necessary. The next one that we're looking at is how. Well, I think I have to talk about this, but yeah, basically 2 minutes, right? Okay, now let's look at how pH affects on enzyme. So similar with temperature, different enzymes have a different pH optimal range, right? So certain enzymes can survive at a very acidic environment, and certain enzymes can survive at a very basic environment. So think of an example, right? I'm gonna see, Lani, can you think of an enzyme in your body, anywhere in your body, where it's located, that it can survive a high acidic environment? Where do you think in your body is a high? Yeah, essentially. In your stomach, you make something known as pepsin, right? It's an enzyme needed to break down proteins. That enzyme is able to survive at a pH range of 2 and above because it needs to have a function in the stomach, right? So the optimal pH is where it functions at best, and depending on where the enzyme works, that can really change. So usually the range can really depend on the enzyme itself, and anytime the enzyme is exposed to a pH that is outside of its range, it immediately denatures. Whether it's acidic or basic, it will cause it to denature. So that's one difference compared to temperature. There is no freezing that's going to occur here. Either if it's too acidic or either if it's too basic, it will cause the enzyme to experience denaturing, which is losing its shape, eventually losing its function. So you can start to graph it out like this. You can see here that different enzymes here have different pH ranges. So pepsin, it prefers to be in a very acidic environment. Trypsin prefers to be in a more basic environment. And urease prefers to be in a very neutral environment. So remember that the range is not fixed for all enzymes. Sorry, not all enzymes have a same range. Each of them will have a specific range in terms of that optimal pH, and you need to be able to remember that. So beyond that range, regardless of whether acidic or basic, it will cause denaturing. The shape will change. Any questions about this one? All right, the next one is talking about substrate and enzyme concentration and how that can affect enzyme activity. So essentially, remember that enzymes require substrates to break it down into products, right? So if there is a lot of enzyme but very little substrate, it's quite pointless because enzymes need substrate to break it down into products. So essentially, the reaction rates are tied together where the reaction rate of enzymes will increase until it eventually reaches a point where we call it saturation point. This is where the reaction rate will stagnate. This means that every enzyme is binding to all the substrate. So every enzyme is occupied binding to substrate. So there isn't any more free enzymes to bind to any more substrates, right? So you can see here, let me try to use the picture to describe it better. Okay, so at the starting rate, there is a higher number of enzymes than substrate. So the reaction rate is very low. That's signified based on the concentration of the substrate given here. Does everyone know molar? Who here does not know the definition of moles? If I say 0.1 moles, do you know what that means? Okay, basically think of that as a concentration measure. The higher the moles, the more concentrated something is, okay? So 0.1 moles means low concentration of substrate, so the reaction rate is low, relatively low. But if we start to increase the amount of substrate within a solution, you can see that the reaction rate is suddenly going up. Essentially, when you increase the substrate concentration, you allow for more binding of enzymes to occur, and eventually you reach a point where every enzyme is occupied. Every enzyme can bind to a substrate. That's why it's called a saturation point. At that point, no free enzymes are available to bind the substrate, and every enzyme will be occupied at every point binding to substrates. That's why it's fixed already after it reaches that point. So you can see here, there are still free enzymes at this point, but as soon as it reaches 1 molar, you can see all the enzymes are occupied and there are free substrate that are floating around. So at this point, no matter what you do, there won't be an increase in the rate of reaction because every enzyme is occupied and they won't be able to bind until they are done breaking that substrate down. It's the same thing with enzyme concentration as well. So with this picture, yeah. So essentially, it would have the same effect with enzyme concentration, right? So if you have a lot of substrate, but a lot of very little amount of enzymes, the reaction rate is going to be very low. So you can add more enzymes to that to increase the rate of reaction, but eventually you will reach something known as a saturation point where it will stagnate and stay constant. So both of them will experience that saturation point because if one or the other is lacking, it won't be able to increase the rate of reaction. They both need to bind to each other to undergo the reaction. So if one of them is even lacking, either the enzyme or the substrate, then the reactions rate will stagnate. Anyone confused about this? Okay, this one is essentially thinking of, okay, I'll try to think of an analogy for this, but essentially every time an enzyme needs to bind to a substrate, if it is already available, then the reaction rate is going to stagnate. If they are free floating, that means there's potential for it to grow. Okay. Next is talking about inhibitors. Okay. This is how a specific molecule can bind to the enzyme and inhibit its function. So think of the repressor proteins that I talked about binding to the operator and repressing the expression of the gene. Similarly, in this case, we have an inhibitor protein that binds to the enzyme to inhibit its function. And there's two types of inhibition that can occur. One is referred to as a competitive inhibition. The other one is referred to as a non-competitive inhibition. This talks about where the molecule binds and how it works. So let's look at each of them. Essentially, enzyme inhibitors are molecules that bind to the enzyme and they also prevent it from catalyzing biochemical reactions. They only specifically inhibit the catalyst of biochemical reactions. They don't enhance at all. Okay, make sure you remember that. These inhibitors are specifically about inhibiting. Okay, so first thing, let's talk about non-competitive inhibition, right? This is essentially when an inhibitor binds to the active site of an enzyme and blocks a substrate from binding onto it. So that's why it's competitive, because it's essentially competing with the substrate to bind to the active site. So that's why we refer to it as competitive inhibition. The other one is non-competitive, because the inhibitor will bind to a different site on the enzyme, and we refer to that as an allosteric site to prevent it from allowing the substrate to bind. That has to do with how it changes the active site once it binds to the enzyme. So I'll go into detail about it in a bit, but remember that the reason why we call it competitive and non-competitive is essentially talking about where the inhibitor binds. We call it competitive because the inhibitor is competing with the substrate to bind to the active site. It's non-competitive because the inhibitor doesn't compete with anything else. It can just bind to the allosteric site whenever it needs to. There's no fuss. That's why it's non-competitive inhibition. So you guys can use the phrases itself to know which one they're talking about. Competitive is always going to be about binding to the active site, and non-competitive is anywhere but the active site. Okay, so let's talk about the first one. Yeah. So competitive inhibition is basically when the competitive inhibitor binds to the active site of the enzyme, and it prevents the substrate from binding to the active site anymore, thus preventing the enzyme from working. It's quite straightforward. It binds to the active site, acts as a block, then the substrate won't be able to bind to that anymore. Non-competitive inhibition is basically when a non-competitive inhibitor binds to the allosteric site, and then that causes a conformational change of the enzyme to then close up its active site. So one is just binding to the active site and blocking it. The other one is causing a conformational change to prevent a substrate from binding. Okay, remember that non-competitive is going to do a conformational change. This one is just binding to the active site and preventing another substrate from binding to it. Anyone confused about this? Okay, hopefully not. Okay, so this inhibition can also be permanent or reversible. So that will depend on the strength of the bonds between the inhibitor and the enzyme. So when it comes to reversible inhibition, that only occurs when the bonds between the inhibitor molecules and enzymes are weak, right? Essentially, if the bonds between them are weak, it can be broken and then substrate can continue binding to it. But if there's irreversible inhibition where the bonds formed between the inhibitor are so strong, then it's essentially never coming off no matter what. So whether that inhibition is broken or not depends highly on the bond strength and also the substrate concentration. If there's a high substrate concentration and the bonds are weak, then it has a high chance of being reversed, right? You can see here what happens, right? So we have a reversible inhibitor, binds to the active site, prevents any more action of the enzyme. So first thing I want to ask, what kind of inhibition is this? Competitive or non-competitive? Can I get Cyril? Okay, this kind of inhibition, is it competitive or non-competitive? Competitive, because it's essentially competing to bind to the active site, so we call it competitive inhibition. So when we're talking about reversible and non-reversible, we are mainly talking about competitive inhibition. So always remember that. So first situation where it's reversible, we have the inhibitor molecule binding to the active site, and then when there is a high amount of substrate present, what happens is the bonds between the inhibitor molecule and the enzyme are eventually broken, and then the substrate continues binding to it. But when we talk about irreversible inhibition, it is no matter how much substrate, no matter how high the substrate concentration is, the inhibitor bonds between the enzyme and the inhibitor molecule are so strong that they can never be broken down, and then that causes that in turn prevents the enzyme from working. So that's why it's referred to as irreversible inhibition. So one is reversible because there's a lot of substrate. The other one is irreversible regardless of how much substrate, and that essentially is because of the bonds that they form. Any questions about these? Yeah, I know. Okay. So let's move on. All right. So essentially, what happens when these inhibitions happen, right? Because enzymes are involved in sometimes they are involved in a biochemical pathway, right? So essentially what happens, we have carbohydrates, and then we have salivary amino acids. Let's you guys don't need to know these names and stuff. I'm just using this as an example for okay, so this is a biochemical pathway, right? It's talking about how enzymes are working together, different enzymes are working together to break down one molecule into its simplest molecule, right? For example, carbohydrates, multiple enzymes work together to eventually get it down to glucose, right? For example, salivary amylase, maltase, and so on. Okay, this is just one pathway. So what happens when one of these enzymes are then inhibited? What do you think? Okay, let's say if salivary amylase is being inhibited, what would happen to this biochemical pathway, ma'am? What do you think? So this is step one, it needs to be broken down into maltose. Step two is that it needs to be broken down into glucose. These steps are carried out by these enzymes, they're facilitated by these enzymes. So what happens when this enzyme is then blocked? It's been inhibited. What do you think will happen to the biochemical pathway? Hmm. So this enzyme is blocked, is inhibited. What will happen to the rest of the pathway? Yeah, essentially, if you can't take step one, no way you can get to step two, right? That is what happens to biochemical pathways when one step of the biochemical pathway is been inhibited. Because at every step, the product formed from the reaction will serve as a substrate for the next step, right? So carbohydrates, that's the substrate. This is the enzyme. The product formed is maltose. But maltose is a product for this one. It acts as the substrate for this one. Do you understand? Substrate is the reactant. Product is what is formed after the chemical reaction. So at every point in a biochemical pathway, the product that is formed will act as the substrate for the next biochemical reaction, right? So essentially, if carbs, then chemical reaction, so I mean, it becomes maltose, right? So this is the substrate, that is the product. What happens so if this is step one, step two in the pathway is this substrate will then become, this product will then become the next substrate to become a different product. Become glucose. So that's why a biochemical pathway can be easily regulated through inhibition, because as soon as one step is blocked, this product isn't going to be formed anymore. So there's no more substrate supplying the second step. That's why the entire thing crumbles and will be brought to a stop, because if the next substrate cannot be formed, then the next reaction won't happen. Then everything else that happens beyond that will also be stopped. Is anyone confused about how this works? Okay. So essentially, the best way you can think about it, if you're going up the stairs, you can't get to step 10 without going from step 1 to 9, right? You can't jump all the way to step 10. It's the same thing with a biochemical pathway. You can't get the final product if you don't get the substrate in between. And if even one step in between them if even one enzyme in this step is being inhibited, then the next substrate won't form, and the whole pathway has been essentially blocked. All right? Anyone confused about this? So this can actually be a good thing sometimes, because essentially a biochemical pathway can regulate itself as well using the products that it makes. So imagine this is one biochemical pathway, right? Sometimes the end product, the glucose here, can act as the inhibiting molecule for a upstream enzyme. So salivary amylase here, this glucose could sometimes act as a inhibiting molecule to prevent salivary amylase from working, because essentially it's saying we've made enough glucose, time to stop this process. Remember how in biology there are negative feedback loops, right? We talked about this, that close off the system by basically regulating itself. That's what's happening here as well. So substrate 1 makes substrate 2, substrate 2 makes substrate 3, substrate 3 makes the final product, and then that final product will inhibit enzyme 1 again, closing off the loop. It is a negative feedback loop, and this is very common in biology. You'll see it all the time. Another one of the examples is in the inhibition of biochemical pathways. So, okay, we need to pick up pace one more time. Last one, coenzymes. This is the last factor that can influence enzyme activity. This one is when a specific partner joins together with the enzyme to allow it to actually carry out its function. So this partner will supply the reaction with either the molecule it needs or a set amount of energy for the reaction to occur. More often than not, it's more about the energy. So think of enzymes as being a structure, and coenzymes supplied with energy, and then the substrate can then be broken down into its product. The coenzyme is also then used up to make that. So you can see this picture here, down here. Yeah. So the enzyme needs a coenzyme to bind to it first, and then only the substrate can bind, allowing the reaction to happen. And the coenzyme is essentially used up for the reaction. So that can either be energy or the molecule, but you guys will more often know it as energy. So essentially, that coenzyme has to be recharged every time they are used, because they're being used up. So think of it as a battery, right? Every time an enzyme needs to carry out its function, a battery needs to supply it with energy and be used up. And that battery then needs to be recharged after that to be used again for this process. So that's what happens actually. It's basically charging and recharging itself every time to allow for this reaction to happen. So that's referred to as coenzyme cycling. So that happens through the action of ATP. Who here has heard of the phrase ATP before? Adenosine triphosphate. So you would have learned about it over the years from year 9 onwards, or in other units as well. ATP is basically the energy currency of life. All life uses energy in the form of ATP, and enzymes also use that energy to catalyze reactions. So the coenzyme here is the ATP molecule. That is what transfers the energy for an enzyme to function, and that is the important coenzyme needed. So that coenzyme, that ATP molecule, donates the energy required for a enzyme to catalyze a reaction, And this is transferred by losing a phosphate group, thus causing it to become a adenosine diphosphate. So essentially what the molecule looks like is this, right? So this is what the molecule looks like when it's charged. It's called ATP, adenosine triphosphate, because there's 3 phosphates. And that's when it has the energy ready to be used. Okay. This coenzyme then binds to the enzyme, releases that energy by giving away that one phosphate group, becoming adenosine diphosphate. Remember di and tri. These suffixes are referring to the number of phosphates, right? Triphosphate is when it has energy. Diphosphate is when it's given the energy away. Okay, once it's given the energy away, it then goes through a process of getting back that phosphate group to become recharged. Remember, like a battery needs to be used and then recharged so that you can use it again. Same thing with adenosine triphosphate. We need to use it for its charge, for its energy, and then once it's used, we need to recharge it so that it becomes useful again. So yeah, essentially this is what happens, right? Adenosine diphosphate becomes adenosine diphosphate when it releases that energy, and that process is referred to as dephosphorylation reaction. And when the adenosine diphosphate needs to be recharged, we refer to it as energy loaded. It's going through something known as a phosphorylation reaction. Okay, so this happens thousands of times every day, right? This one molecule, adenosine diphosphate, acts as a coenzyme to bind to a enzyme, releases the energy for the substrate to be broken down into product, and then the ATP gets recharged to become, the ADP becomes recharged to become ATP again, and then carries it out all over again. So this process is continuous for thousands of times because it needs to keep supplying energy to the enzyme so that it can undergo the process of catalyzing the reaction. So that's pretty much everything to do with the factors affecting enzyme activity. Remember that coenzymes, you guys will basically be thinking about ATP mainly. So ATP is energy. So coenzymes, think of it as supplying energy to carry out the catalytic activity of enzymes, and they do it by binding to the enzyme first, allowing the substrate to bind to that, then only undergoing the process. Okay, so it's very important that you know that the coenzyme binds first, then only allows the substrate to bind, and then it uses up the coenzyme to break down the products. And that coenzyme, we refer to it as adenosine triphosphate. So adenosine triphosphate is just one of the coenzymes out there, but that's the one that you guys have to really focus on for your studies. It supplies energy to the enzymes to carry out the catalyst reaction, and after it's supplied that energy, it will be released and will get recharged to become ATP again, undergoing the same process over and over. Any questions about this?